Preparatory model question paper 17, plant pathology MCQ based. I'm NH Shankar Reddy, PhD plant pathology. So first one is late blight of potato is caused by Pseudomonas solanacearum, Alternaria solani, Pythium aphanidermatum, and Phytophthora infestans. We know very well the main reason for the initiation of a new branch, that is plant pathology, is due to late blight of potato only, which is caused by Phytophthora infestans. The life cycle of the pathogen was discovered by Anton de Berry. He is considered as father of plant pathology. And the name Phytophthora infestans is also given by Anton de Berry only. Phyto means plant, Thora means destroyer. Phytophthora means completely a plant destroyer. Generally, the Pseudomonas solanacearum generally causes bacterial diseases. Alternaria solani, Alternaria kind of diseases like some kind, if you see vegetables and other kinds of diseases. And if you see this pythium damping of generally causes damping of diseases. The next one is who demonstrated gene for gene hypothesis concept in 1946. This is a very important concept, gene for gene hypothesis, which was given by H.H. Floor in 1946. Muller and Borger they discovered this phytoalexins. And next one is Van der Planck, considered as father of plant disease epidemiology. And the third one is just forget about the third one because the third and fourth are same. The next one is fungi and diseases in plants was written by. So here there are two books are there which actually confuses us: fungi and diseases in plants, fungi and plant disease. Actually fungi and diseases in plants was written by E.J. Butler in 1918, whereas fungi and plant disease was written by B.B. Munkur in 1948, not 1988, it's 1948. I repeat, fungi and diseases in plants was written by E.J. Butler, whereas fungi and plant disease was written by B.B. Munkur. Anton de Berry, he also wrote a book called Comparative Morphology and Biology of the Fungi, Mycetosia and Bacteria. And Robert Hartig is generally considered father of forest plant pathology. And black leg of potato is generally caused by, actually this black leg of potato is caused by fungi. Generally fomalingum is also many cases it is reported that it is involved in the black leg of potato, but since in the options they have given only bacterial pathogens. Irvinia amylovora generally causes soft rots, Agrobacterium tumefaciens generally causes galls, Pseudomonas solanacearum bacterial wilts, and then Irvinia keratovora. This Irvinia keratovora also causes soft rot and is also responsible for this black leg of potato. So the answer is Irvinia keratovora. So the next one is the variability of bacteria can be achieved through. So generally variability in the sense just changes are creating the changes in bacteria generally can be achieved through. So conjugation, transformation, transduction. So these three are sexual recombinations or sexual reproduction types in bacteria. These three can create variations in bacteria. So the answer is all the above. For our understanding, I have given a small table here. It will be very easy to remember other concepts as well. Generally, conjugation was discovered in organism Escherichia coli, discovered by Lederberg and Tatum in the year 1946. Transformation was discovered by pneumonia, discovered by Griffith et al. In 1928. Transduction was discovered in the organism Salmonella, discovered by Lederberg and Zinder in the year of 1952. This table will tell everything about this bacterial recombinations. So in which organism the recombination is discovered, who discovered, the year of discovery, everything was simplified in this table. So the next one is the bacterium with a single flagellum at both ends. They are telling single flagellum at the both ends. To understand this, first of all, we will go to the bacterial flagellation. So generally flagella is the locomotor organ, right? There are a couple of types of flagella in bacteria. If you see A-trichus means no trichus means hair, okay? So A means no hair. Flagella contains no hair or flagella is absent. So sorry, generally no flagella or flagella is absent, that is called A-trichus. Example is xylella. Second one is monotrichus or polar flagellum. Mono means single. So this is A-trichus. Mono means single. A single flagellum at one side of the bacterium, any one side, maybe right or left. Example is Xanthomonas. This is a diagrammatic representation. Amphitrichus, a single flagellum at both ends. So one flagellum at both ends, that is called Amphitrichus. Pseudomonas species. This is the example. So here the next one is a peritrichus. Peritrichus means flagella all over the body. So all over the body the flagella is present. So example is Irvinia, Pantovia, Escherichia coli and Clostridium will produce this kind of flagellation. So the next one is the lopotrichotes, tufts of flagella to both poles. If you see, there are plenty number of flagella, tufts of flagella, more than 2 to 3 types of flagella, more than 2 to 3 flagella is present in both sides. Example is spirulina. Next one is cephalotrichus, groups of flagella to one side. Example is pseudomonas. Only a group of flagella is present at only one side, that is called cephalotrichus. Example is pseudomonas fluorescens. The next one is a circumtracheal or polar flagella, so which means bacteria with the lateral flagella on one or both sides, sometimes one side or both side. Example is agrobacterium, general agrobacterium tumefaciens. So these are all the flagellation types in bacteria. So let's see the question. A bacteria with a single flagellum at both ends, single flagellum at both ends, the example is amphitrichus. So the answer is amphitrichus. The next one is a bacteria with a single flagellum, same amphitrichus only. The next one is a solar heat treatment used for the management or control of seed gall, black rust, loose mud and brown rust. Solar heat treatment is generally recommended for wheat loose mud, Histogonoida triticale. The next one is a citrus greening disease is mainly confined to West India, East India, South India, North India. Generally, the citrus greening is more severe and more especially present at North India only because of temperature. The next one is a common scab of potato can be caused by the pathogen. Streptomyces scabis, Streptomyces aureofaciens, Streptomyces graecosus, Streptomyces fluviaceus. See, generally this common scab is caused by Streptomyces scabis, whereas the Streptomyces graecosus is an antibiotic pathogen where streptomycin is produced from. Next one is the citrus canker, which originated from China, is caused by Albugo candida, which is actually a fungal pathogen, Irvinia amylovara, which causes fire blight of apple, and Claviceps fusiformis, again generally causes ergot diseases. So the answer is Xanthomonas axonopodis, pathovirus generally causes citrus canker in originated from China. Next one is yellow ear rot of wheat caused by Clavibacter triticale is usually associated with the which nematode? So this is yellow ear rot is actually a bacterial disease. Generally it associated with a nematode that is called, which one they are asking, Heterodera avinae, Anguna triticale, Meldogan incognita, Paratrianchulus. So the answer is Anguna triticale. Generally this is a very important disease when it comes to wheat. This is actually associated with a nematode that is Anguna triticale, and this Anguna triticale is the first plant parasitic nematode to be discovered by Needham 1743. The next one is which one is a sexual spore? Sporangiospore, chlamydospore, zygospore, zoospore. This sporangiospore, chlamydospores and zoospores are the asexual spore. Now ultimate answer is zygospore. Next one, how many ascospore packed inside an asci? Every ascus in ascomycota group of contains 8 number of ascospore. Every basidium in basidiomycota contains 4 number of basidiospore. So here ascomycota group of fungi contains 8 number of ascospores. The sexual fruiting body of ascomycota without hostule. So generally perithecium, clistothecium, apothecium, perithecium. Perithecium is hostile ascocarp. Apothecium is the opened one, right? So clistothecium contains no such kind of opening, that is hostile. So the answer is clistothecium. Clistothecium is a completely closed ascocarp. Apothecium is a completely opened. So perithecium is partially opened with a small slit or opening is known as ostiole. What they asked is without ostiole. So the answer is clistothecium. Next one is Albugo candida producing the sexual spores, was known as. First of all, we understand that this Albugo candida is a oomycetes group of pathogen, generally causes white rust, right? So here basidiospores, not at all, because it's a basidiomycota. Zoospores, generally they asked sexual spores. Of course zoospores are belonging to this oomycetes, but zoospores are asexual spores. Oospore, ascospore. Ascospore is ascomycota. Since it is a oomycetes group of fungi, oomycetes produces a sexual spore is a oospore, asexual spore is a zoospore. So the answer is they asked a sexual spore, so the answer is a oospore. Next one, downy mildew disease of pale millet is a primarily a seed-borne, soil-borne, airborne, waterborne. Generally this is a primary pale millet of downy mildew of pale millet is generally a soil-borne pathogen primarily. Second one is stem gall of coriander is caused, but it's a very important when it comes to coriander. Plasmapara viticola generally causes downy mildews. Peronospora pisci, maybe downy mildews in peas, not sure. Next one is a Protomyces macrosporus. So the answer is a Protomyces macrosporus, stem gall of coriander pathogen. Loose mud of wheat is a internally seed-borne, externally seed-borne, not defined, A and B. So the best example of internally seed-borne pathogen is a wheat loose mud. So the answer is A. Next one, ergot of rye is caused by Claviceps fusiformis, Claviceps purpurea, Claviceps sativa, and all the above. Actually, generally this ergot, if you see, two kinds of pathogens are generally mentioned, like Claviceps fusiformis and Claviceps purpurea. Both generally in basic, this ergot is mainly caused by this Claviceps purpurea only. Next one is which fungicide used for the control of cover smut. So for all smut diseases we can recommend Vitavax. For all rust diseases we can recommend plant vax. So that we already discussed, right? So externally seed-borne fungi of grain smut of sorghum is caused by the pathogen. First of all, you forgot about all those things. They are asking just pathogen name. Grain smut is caused by Sporisorium sorgi or Spatialotheca sorgi. Spatialotheca reliana, Spatialotheca cruenta, Spatialotheca sorgi, Toliposporium ehrenbergi. So the Spatialotheca sorgi causes generally grain smut. Okay, so the answer is a grain smut. Here for our understanding I have mentioned a couple of things here. Grain smut, head smut, loose smut, long smut. Grain smut is caused by Spatialotheca sorgi, now name changed to Sporisorium sorgi. Head smut is caused by Spatialotheca reliana, the name changed into Sporisorium reliana. Loose smut, Spatialotheca cruenta, now name changed to Sporisorium cruenta. Long smut, Toliposporium ehrenbergi, now name changed to Sporisorium ehrenbergi. The site of infection of grain smut is generally ovary, whereas ovary for grain smut, loose smut, and long smut. For when it comes to head smut, it is inflorescence. Disease survival, generally this grain smut survive as externally seed-borne pathogen, whereas head smut survive as a soil and seed-borne. Again, this loose smut is also externally seed-borne, long smut is a airborne. Type survival is generally 10 years when it comes to grain smut, more than 10 years, in between 10 to 13 years. Head smut is 2 years, whereas a long smut is 4 years. Sorry, loose smut is 4 years, long smut is 2 years. This table is very important. If we understand this table, we can answer the questions very easily. Next one, Mitra was the first reported a kernel bunt of wheat from a kernel in the year 1931. This kernel bunt of wheat was reported. So this Mitra named this kernel bunt as Tilletia indica. Later, BB Munkur name changed to Neovesia indica. Okay, now if we see the literature, both Tilletia indica and Neovesia indica will be available. So now we can use this both names, but generally Neovesia indica was given by Munkur. Nowadays it was following most of the literature it appears as Neovesia indica, but later in the conferences they told that both names are applicable, but anyone is right. The kernel bunt of wheat producing a foul smell odor in the field is due to trimethylamine, tetramethylamine, diallyl propylamine, allyl propylamine. So the answer is trimethylamine. Due to production of trimethylamine only, it will produce fish odor. So the next one is yellow wheat yellow rust is caused by Puccinia graminestritis, Puccinia striformis, Puccinia regonita. So generally this yellow rust is Puccinia striformis. Black rust is generally black rust or stem rust, Puccinia graminestritis. Yellow rust means Puccinia striformis. Brown rust or leaf rust means Puccinia regonita. Next one is flax and linseed rust can be controlled by, again all kinds of rust can be controlled by flax and linseed rust, right? So generally rust can be controlled by application of our seed treatment with the plant wax. Smut can be recommended by Vitavax. So they have mentioned other things, Agrosan, Serasan, Vitavax and Borax. So among that Borax can be recommended for this flaxseed and linseed rust. The next one is Coltrotrichum falcatum producing asexual fruiting body. This Coltrotrichum falcatum causes a red rot of sugarcane, first of all. So this produces ASRB. This Coltrotrichum generally produces, normally if we say the Coltrotrichum, it produces depressed spots. Asexual fruiting body is ASRB, and the sexual spores it will produce is generally ascospores. Asexual spores are conidiopores, where this ASRB produces this conidiospores. Next one, PGNP can be intercropped with the dash crop for the effective management of the disease. Generally, this PGNP intercropped with the sorghum for the effective management of this PGNP, various kinds of PGNP diseases, and especially this wilted disease. Citrus greening is caused by Candidatus Liberibacter asiaticus, generally the citrus greening. The citrus greening, otherwise called as horned long pink disease, right? So generally it is caused by fastidious vascular bacteria or FAB. The next one, Puccinia graminis producing iridospores are disseminated by, generally iridospore disseminated by wind, right? Fungi imperfect is related to, or dustbin fungi generally related to Oomycota, Basidiomycota, Ascomycota are, if you see Ascomycota and Basidiomycota are higher fungi, developed fungi. This is Oomycota is also almost fungal-like resemblance. But when it comes to this Deuteromycota, it is called fungi imperfecti or dustbin fungi. Dustbin fungi was given by Fackel, whereas C.T. Ingold named this Deuteromycota fungi as a fungi imperfecti. C.T. Ingold in 1971. Okay, why it is called a dustbin fungi or fungi imperfecti? Because it produces no sexual life cycle. Sorry, sexual life cycle. That's why it's called as fungi imperfecti. So the next one is downy mildew is actually related to oomycetes, zygomycetes, chytridiomycetes, hypocytridiomycetes. We know very well all this downy mildews, damping ups comes under this oomycetes group of pathogen. So the answer is oomycetes. Which of the following scientists received the Borlaug Award in 2005? C.D. Mai, V.L. Chopra, Ratan Lal and S. Nagarajan. So the answer is S. Nagarajan received the Borlaug Award in 2005. So the next one is micrografting is used for the management of our DAS-free plants. RLO, MLO, virus, bacteria. I think virus-free plants, micrografting is generally recommended for. The next one, Panama disease of banana can be managed by. First of all, we need to know Panama disease or Fusarium wilt of banana is caused by Fusarium oxysporum pharma species cubensis. There are 4 races are there, right? TR1, tropical race 1, TR2, TR3, TR4. Nowadays TR4 is very dangerous. In recently, James Dale, an Australian scientist, developed a banana variety that is resistant to this Sigatoka leaf spot, and he was extensively working on this TR race 4. Actually, I met this James Dale in 2020 in Banana International Conference at Tiruchirappalli. I think that happens in February 22 to 25. I met there this scientist, and we just had a we just spoke something about TR4. Since even though I understood nothing about TR4, but I just had a very good talk about this James Dale. So he developed now he is actually working on TR4 tropical race 4. So coming to this point, Panama disease of banana is actually managed by spraying of zinc carrier, spraying of copper sulfate, application of lime, provide adequate irrigation. Generally, application of lime is more than enough to manage this disease very effectively. Sometimes this disease is also associated with banana burrowing nematode, Rhadophilus similis. Okay, so this application of lime itself is the best management practices. Just changing the pH is it is the best management practice. So the next one is which penicillium species used for the cheese making? So to understand this, first of all, we will go here. First of all, there are a couple of common things are there. Aspergillus. So in Aspergillus itself, there are a couple of species are there. If you see black mold Aspergillus niger, green mold Aspergillus flavus, white mold Aspergillus candida, brown mold Aspergillus tamari, yellow mold Aspergillus acarius. These are all the Aspergillus species, right? Let's go to penicillium. Blue cheese fungus Penicillium roqueforti, or cheese making fungus Penicillium roqueforti. Blue mold fungus Penicillium italicum. Green mold fungus Penicillium digitatum. Rot of apple, which is caused by Penicillium expansion. Generally, it causes a rot of apples. In citrus, we can also see in apples. Also, we can see some green color outgrowth, right? So that is generally rot of apples or citrus. This is caused by Penicillium expansion. In general, gray mold fungus means Botrytis cinerea. Black mold fungus means Rhizophus stolonifer. If they mentioned the black mold fungus of Aspergillus, then we can go for Aspergillus niger. If they have not mentioned anything, generally the black mold fungus means Rhizophus stolonifer. White mold fungus Sclerotinia, Sclerotiarum. Last one is peat mold fungus, species Astrocoderma. These are all the various kinds of molds or funguses. So the question is, which penicillium species used for cheese making? So we have already seen the cheese making fungus is Penicillium roqueforti. Penicillium notatum, now it is called as Penicillium chrysogenum. The name changed to Penicillium chrysogenum. This Penicillium chrysogenum was given by Thom 1904, where this penicillin is actually isolated from, right? The next one is the ELISA plate is made up of. ELISA means enzyme linked immunosorbent assay. The technique was developed by Clark and Adam 1977. This plate is actually made up of para-nitrophenyl phosphate, that is PNPP, para-nitrophenyl phosphate. To have a better understanding about the ELISA, I have given a couple of some questions here. the most extremely used sensitive technique for the detection of plant pathogens or plant viruses, ELISA. What is the enzyme used in ELISA? Horseradish peroxidase and alkaline phosphatase. Next one, what is the substrate used in ELISA? Substrate used in ELISA is a para-nitrophenyl phosphate and a TMB, tetratimethylbenzene. Okay. Next one, who coined the term ELISA? The term ELISA was coined by Eva Engel and Peter Pellman in 1971. Who made the ELISA plate? So ELISA plate is actually made by Guy Takashi 1951. Sensitivity of ELISA is 1 to 10 nanogram. Sensitivity of PCR is 1 to 10 picogram. PCR, like that you can remember. Plate of ELISA is made up of polystyrene plate generally. So here I think I have told you a para-nitrophenyl phosphate. It is supposed to be ELISA contains a substrate, not ELISA plate is made up of. So the question is supposed to be ELISA substrate. ELISA contains a substrate para-nitrophenyl phosphate. Generally ELISA plate is made up of a polystyrene plate. Okay, so this question itself is wrong. So the next one is the most sensitive, that is again ELISA only. Generally ELISA contains 96 wells. There are other wells also, 2, 24, 48, 96, 384, various kinds of wells are available. But the most commonly used ELISA plate is made up of polystyrene and it contains 96 wells, 12 into 8. Pore size of ELISA, each pore size, each hole size or each pore size of ELISA is 0.4 micrometer or microliter. I think it's not micrometer, microliter. The next one is, what is the substrate used in PCR, that is for amplification? Tag polymerase, nitrocellulose, para-nitrophenyl phosphate, nitro blue tetrazoline. So the is tag polymerase. Tag polymerase DNA, that is tag polymerase thermus aquaticus, right? So that holds the DNA at high temperatures. So that's why we used this substrate tag polymerase. Second one is RFL, restriction fragment length polymorphism. Next one is RNA interference or PTGS was first discovered in the organism. So this RNA interference or PTGS was first discovered in the organism nematode Caenorhabdis elegans, right? That was discovered by Andrew Fire and Craig Mello. Next one, parasexuality was discovered first in the organism. So this parasexuality was first discovered by Ponticarvo and Roper in 1952. They discovered an organism called Aspergillus nidulans. Aspergillus fumigatus, nigricans, parallel. So the answer is Aspergillus nidulans here. Next one is a pyriform shaped conidia is produced by, so Coltotrichum alternaria curvularia. Alternaria produce bell shaped, Coltotrichum oil globule. The next one is a curvularia curve shaped. The next one almost there is only one thing left, that is a pyricularia, which produces pyriform shaped conidia or pear shaped conidia. 2 septations and 3 celled. The conidia of pyricularia is okay. Flame shaped conidia is produced by curvularia, claviceps, sclerospora, venturia. So the venturia generally produces flame shaped conidia. Which one is a dead man's finger? Xylaria polymorpha, Xylaria hypoxylan, Serpulolacrimens, Rhizoctonia solani. This Rhizoctonia solani is sheath blight. We know very well. This Xylaria polymorpha is a dead man finger. So this Xylaria hypoxylan is candle snuff fungi, I think. Serpulolacrimens is a household fungus or house fungus. The next question is example of house fungus. The same thing. Serpulolacrimens. And Nigrospora varese generally produces a jet black conidia. Okay, the next question itself is same. Jet black conidia is produced by Magnoporthia varese, Rhizoctonia solani, Nigrospora varese, all the above. So the answer is the Nigrospora varese produces a jet black conidia. So to have a better understanding about this conidia, on the next slide I have placed a couple of conidias and as well as their shapes and colors and all those things. Which pathogen enters through stomata? Bacterial blight, black rot of cabbage, potato scab. Now we will see black rot of cabbage enters through hydrothodes and bacterial leaf blight of rice also. Then stomata enters through this bacterial leaf streak pathogen can enter through this stomata. So the answer is bacterial leaf streak, D. Now I told you different shapes of conidia, right? Here we can see sickle shaped conidia is produced by Fusarium and Coltotrichum. Both produces sickle shaped conidia or half crescent, half moon shaped conidias. Okay. Flame shaped conidia is produced by apple scab produces a flame shaped conidia. Mushroom shaped conidia is produced by Claviceps fusiformis. Rhomboidal shaped conidia is produced by Phylloctena. Next one, vermiform or multicellular conidia is produced by Cercospora and Cercosporidium. Pyriform conidia or pear shaped conidia is produced by Pyricularia varese. Muriform conidia or muriform septation kind of conidia can be observed in Alternaria. Piliform conidia is produced by Mycospirilla and Septoria can produce piliform shape of conidia. These are all the different shapes of conidia. Okay. So the next one is this one is different shapes of ascospores. Now we will see. Spindle shaped ascospore is produced by Spermoptera gossypi and Eremothysium cori. Hat shaped ascospore is produced by Ascardia rubensis. Piliform shaped ascospore is produced by Claviceps. Piliform shaped ascospore is again one more pathogen is also that produces piliform shaped, that is a Cochleobolus. Here this piliform is long and narrow, it is a little bit bulged, right? And the next one is a boat shaped or footprint shaped ascospore is produced by Venturia inequalis. Slipper shaped ascospore is produced by Pleospora. These are all the different shapes of ascospores. So the next question is which pathogen enter through lent cells? Potato scab, citrus canker, bacterial canker of tomato, all the above. So the answer is bacterial canker of tomato can enter into the plant cells through lent cells. Next one is Indian stem rust rules was given by. So there are two kinds of things are related to wheat stem rust: Indian stem rust rules and Paxinia pathway. Indian stem rust rules was given by Nagarajan and Singh, whereas Paxinia pathway was given by Nagarajan and Joshi. Here the question is the Indian stem rust rules, Nagarajan and Singh, that was given by Nagarajan and Singh 75, whereas the Paxinia pathway was given by Nagarajan and Joshi in 1980. So the next one is expansion of AUDPC. AUDPC, the concept of this AUDPC was again given by J.E. Vanderplank, is the father of plant disease epidemiology. So AUDPC means area under disease progress curve. That is the answer. Next one is Van Everdingen rules or Dutch rules are applicable for. So this Van Everdingen rules, Beemont rules, Dutch rules, Irish rules, all the rules are applicable for late blight of potato. For apple scab, we have one thing that is Mills rules, right? Mills rules applicable for apple scab. Next one is tomato silver leaf is caused by, generally this tomato silver leaf is caused by an alkyl pollutant called peroxyacyl nitrate. So hydrogen fluoride, nitrogen deficient, other things I have given something here. On the left side, name of the air pollutant and what are the hosts are susceptible along with what is the toxicity level. So this table is important, sometimes we may expect. So sulfur dioxide, generally alpha, violet, conifers, peas and cottons and beans are generally susceptible. At 0.3 to 0.5 ppm parts per million it is toxic. Generally nitrogen oxide, beans and tomatoes are susceptible, 2 to 3 ppm. Hydrogen fluoride, it is the most sensitive and most toxic one. It at 0.1 to 0.2 ppb parts per billion itself, it is highly toxic to corn, peach and tulip. Next one, chloride, many kinds of plants, 0.1 ppm. Ozone, tobacco, beans, cereals, alfalfa, petuna, pine, citrus and corn at 0.1 through 0.3. The next one is peroxyacyl nitrate, spinach, petuna, tomato, lettuce and dahlia, and I have mentioned something silver leaf of tomato, which is actually peroxyacyl nitrate, 0.01 to 0.02 ppm. Ethylene, many kinds of plant, 0.05 ppm, it is constant toxic. So they have asked tomato leaf curl is caused by, tomato silver leaf, which is a toxicity due to peroxyacyl nitrate. So the answer is B. Whiptail of cauliflower is due to, it is a very important question, repeated question, molybdenum deficiency. The next one, which one is a zinc deficiency? Little leaf of citrus, rice, Kara disease, white tip of maize, all the above. So the answer is all the above actually. For that I have given a small table here. Rice, Kara, little leaf of apple, grapes and stone fruit, citrus, foliocellulosis, white bud of maize or white tip of corn, yellow streak of maize, all these are caused by the deficiency of zinc. So the answer is all the above actually. The next one, which country have the toughest quarantine standards in the world? Generally Australia is the toughest quarantine standards in the world. So the answer is Australia. Which fungicide recommended for rust disease? Oxycarboxyn, carboxyn, metaloxyl, carbendazim. So the generally fungicide recommended for rust diseases is oxycarboxyn. If it is a smut, carboxyn. Oxycarboxyn trade name is plant wax, carboxyn trade name is Vitavax. Which fungicide used for the control of smut diseases? Just now we discussed carboxyn is generally recommended for the control or management of smut diseases, right? Next one, book written by S.C. Vyas, systemic fungicides in plant disease control, the fungicides. Generally S.C. Vyas wrote a book called Systemic Fungicides. Biocontrol for wheat leaf rust. For rust, whatever it is, a leaf rust or whatever the rust, there is a common fungicide called Darluca pilum. So Darluca pilum. Ampelomyces quisqualis generally recommended for grapes powdery mildew diseases, right? Trichoderma viride, almost all kinds of soil-borne pathogens. Glycolidium variants, maybe some kind of damping of, I'm not sure, probably. And this biocontrol concepts and all those things that was clearly given in the previously published videos, that is biocontrol related topics. So book written by Baker and Cook, Biocontrol of Plant Pathogen, Antibiotics and Plant Disease Control, Systemic Fungicide, Plant Disease Epidemics and Control. So Biocontrol of Plant Pathogens was the book that was written by Baker and Cook in 1983. Antibiotics and Plant Disease Management, Antibiotics and Plant Disease Control was written by M.J. Tirumalacharyya, is considered a father of Indian antibiotics. Systemic Fungicide, the book was written by S.C. Vyas. Plant Disease Epidemics and Control was written by J.E. Vander Planck, 1963. Next one. Green leaf hopper is the primary vector for sheath rot, crescent disease, rice tungro, rhizophyra disease. Rhizophyra is a nematode. Crescent disease means it's bacterial leaf blight. Sheath rot means that is Saraclodium varize. So all are fungal and bacterial nematode diseases. So here rice tungro is the viral diseases. Viral disease, that rice tungro is generally transmitted by green leaf hopper, Nephrotictis virescens, or Nephrotictis syncticipus, Nigropectus, and various kinds of species are there. Generally Nephrotictis virescens is the most recognized one. Viride contains genome, DNA, RNA both, DNA only, RNA only. Generally, almost all kinds of viride contains RNA as a genetic material. The two single-stranded RNA as a genetic material. Not only one viride, all the virides. So far, there have been 40 virides have been discovered. All virides contain single-stranded RNA as a genetic material. Next one, internal chlorosis is due to zinc deficiency, iron deficiency, molybdenum deficiency, boron deficiency. Generally, this chlorosis is normal internal chlorosis generally due to iron deficiency. Stalk rot of maize is caused by Irvinia chrysanthemum pathovarzie, Irvinia chrysanthemum pathovar malus, pathovar avine. So the name itself indicates pathovarzie, host specific, right? So this soft rot is caused by Irvinia chrysanthemum pathovarzie. Black heart of potato is due to high temperature, water deficiency, oxygen deficiency, boron deficiency. These are all nutritional disorders we have already published in the subject specific videos. Though this generally black heart is caused by oxygen deficiency. Next one, inhibitory substance present in the pears which prevent actually fire blood disease. Vitic acid generally component of grapes, right? Malic acid, ascorbic acid. Generally the pears contains malic acid. So the best management practice is if we increase the content of malic acid in the leaves of pear, we can automatically manage this fire blood disease. So the answer is malic acid. Present chairman of ASRB. Actually, this year by ASRB chairman changes year to year. So at 2019 time, AK Mishra was the chairman. At present, again Gurbhajan Singh, I think so. At present, at the time of 2019 is ASRB chairman is AK Mishra. At present, I think it's Gurbhajan Singh. I'm not sure. Probably Gurbhajan Singh. Trilochan Mohapatra is ICAR Director General before. Now ICAR Director General is changed. I think one minister was there. Actually, I forgot his name. He is the present Director General. Recently in video conference also, I just not me, everyone. There are plenty of colleges are involved. We also participated in that. Actually, forgot his name. Central Union Minister, probably Agriculture Minister's name. Something was there. I don't know, probably. But the answer for this is AK Mishra. At present is maybe Gurbhajan Singh. Akiochi is due to sulfur toxicity. In previous also we have seen. Which of the following is a cultivated mushroom? Amanita citrina, Amanita pallidus, Amanita bisporus. All are cultivated mushrooms only. All the above. Hinosan used for powdery mildew, fusarium mildew of tomato, rice brown leaf spot. So this powdery mildews generally we can recommend tridamorph group of fungicide, right? So that is Bileton. So this for rice blast there is a specific fungicide called tricyclozole. Along with the tricyclozole, we can also recommend two other fungicides, edifenfos and iprobenfos. The trade names are Hinozan and one more name is there. But generally, this Hinozan is recommended for rice blast. A disease occurring severely and confined to a small locality or area. Epidemic means sudden outbreak. Endemic means confined to a particular area or locality. Pandemic means present all over the world. Sporadic means it occurs irregularly. So the answer is endemic. The best example for endemic disease is potato wart, Syncytium indiabaticum. Cutter disease of cardamom is transmitted by Mysis persicale, Thrips, Tabaci, Apis gossopi, and Pentaloria nigronervosa. Generally, there are two diseases, banana bunchy top and cutter disease of cardamom. These both are transmitted by black aphid, that is Pentaloria nigronervosa. The next one is citrus stubborn is caused by Spiroplasma concalis, Spiroplasma spectris, Spiroplasma populsona, and Spiroplasma myrum. So this citrus stubborn is generally responsible for, if you see in phytoplasmal disease, first reported phytoplasmal disease, first reported spiroplasmal disease, first reported phytoplasmal disease, mulberry dwarf. First reported spiroplasmal disease constant, right? So such kind of disease will be there. They have mentioned citrus stubborn. So it is a Spiroplasma citri generally. White tip of rice is caused by Flingitis bacillus was first reported by Daster. So Daster was the one who first reported this white tip of nematode, that is Flingitis bacillus. Certain bacteria to gain entry into the host tissues to dissolve cell wall of the plant tissue that it produces pectinolytic enzymes, zebralyc acid, phytotoxins, and pyrrolidin. So here the answer is a pectinolytic enzymes. Sorry, pectinolytic enzymes. These pectinolytic enzymes are very helpful, especially produced by the bacteria, helps to gain the entry into the plant cells. This actually digests the tissues, so that it will helpful to gain the entry, and it will, like once it get enters, it will make all the tissues soft, and water like exudates can be observed. Zebralyc acid generally we can see in it is in a growth hormone that can mainly seen in one rice disease, that is Zibberella fusicorae, which causes Bacana disease of rice, right? Pyrrolidin, that is the toxin produced by a rice blast. Next one is Carathane is effective against. Carathane is a group of fungicide that is generally effective against the powdery mildews. This Carathane group of fungicide especially recommended for this generally powdery mildews. So the next one is citrus disease could be effectively managed by. Generally, there is a technique called cross protection technique in the uh, model paper 16 or maybe 15 also I have discussed. Originally there is a technique called cross protection that is originally developed for TMV, but it is exploited, or nowadays we are exploiting or using against citrus disease virus. So this cross protection technique is very effective against this citrus disease virus. So pan causes which symptom? Pan paraxysalinated, I think we have already discussed. Silver leaf of tomato, right? Spore, sexual spore produced by the fungi causing downy mildew disease. All downy mildew produces sexual spore is a oospore. Asexual spores has a zoospore. So the answer is a oospore. Then next one is a group of carbendazim. Carbendazim belongs to which group they are asking? So benzimidazole, oxazine group of fungicide, strobilurin, heterocyclic nitrogen compounds. For our understanding, easy understanding, I have placed one diagram on the next slide. If we see here, these are the classifications of the fungicide based on the chemical nature. So sulfur, there are two types of sulfur is there: inorganic sulfur and organic sulfur. So powder sulfur, wettable sulfur, lime sulfur comes under inorganic sulfur. Ferbum, zirum, thirum, zinab, menab, nebum, babum, these all are comes under organic sulfur. These organic sulfur fungicides are especially used against seed treatment fungicides. Copper fungicides like Bodo mixture, Burgundy mixture, cuprous oxide, chestnut compound, these are all are copper fungicide. In mercury again there are two types, inorganic and organic. Mercurial chloride and mercurous chloride are comes under inorganic. Phenyl mercury acetate and methoxyethyl mercury acetate are comes under organic. Quinol fungicides are chloralin and dichlorine. I just gave a few examples only. There are hundreds of examples are there. I'm just giving few examples only. Heterocyclic nitrogen compounds, Captan and Folfate are the best examples. Oxazine group of carboxyl and oxycarboxyl. We know very well this carboxyl and oxycarboxyl is the first discovered or developed systemic fungicides, or oxazine group of fungicide, carboxyl and oxycarboxyl, that was developed by Van Schimling and Marshall Kulka 1966. Benzimidazole group of fungicide, benomyl and this carbendazim comes under this benzimidazole group of fungicide. Others like PCNB, dodentine compounds and antibiotics also there. Now they asked carbendazim comes under, right? So this carbendazim comes under benzimidazole group of fungicide. So the answer is A, benzimidazole group of fungicide. Next one, banana burrowing nematoda is a migratory endoparasite, sedentary endoparasite, ectoparasite, endoparasite. For to have an understanding, I have given here some types of parasitisms of various kinds of nematode genera. So Anguna is generally seed gall nematode, which is a migratory endoparasite. Bursa phalangelus, red ring or wilt, migratory endoparasite. Cricosamella, ring nematode, sedentary endoparasite. Ditylenges, stem and bulb nematode, migratory endoparasite. Globodera, golden cyst nematode, sedentary endoparasite. Helicotylenchulus, spiral nematode, which is a migratory ecto and endoparasite. Heterodera cyst nematode, sedentary endoparasite. Hirschmaniella, rice root nematode, migratory endoparasite. Haplolimus lans nematode migratory endoparasite, Malodogene root nematode sedentary endoparasite, Pratylenchulus lesion nematode migratory ectoderm endoparasite, Rhadophilus banana burrowing nematode migratory endoparasite, Rotalenchulus reniform nematode sedentary semi endoparasite, Scutellanema am parasite, am nematode migratory ectoderm endoparasite. What they have asked is a banana burrowing nematode, that is Rhadophilus similars, which is a migratory endoparasite. So the answer is a migratory endoparasite. So the next one is the filter nature of viruses was first demonstrated by. So actually the first one who Mayer thought that it is caused by bacteria. Later it was filtered by Ivanovsky and told that it is not caused by bacteria because the causal organism is smaller than that of bacteria. That was told by Ivanovsky. So this filter nature of virus was given by Ivanovsky only. Okay. So the next one is the hypovirulence is due to. Hypovirulence phenomena generally used for the management of some kind of disease, Dutch elm diseases, or maybe various kinds of diseases. So we can generally use this technique called hypovirulence, just reducing the virulence of the pathogen by using the double stranded RNA viruses. Here the fungal pathogen virulence is reduced by using the viral pathogens, that is especially double stranded RNA. So this hypovirulence phenomena is mainly involved in the double stranded RNA. Next one, white rust causing pathogen, albugo, pythium, puccinia, istulago. Now we will see. All forget about albugo, pythium generally damping of, puccinia rust, istulago smuts. So ultimately the answer is albugo. Albugo causes white rust disease. Next one is American Phytopathology Society was established in the year. It is one of the largest society in the world. American Phytopathological Society established in the year 1908. It is the largest society. But when it comes to oldest society, Netherlands Society of Plant Pathology is the oldest, which was established in 1891. Next one, 1% Bodo mixture contains 1:1:100, 1 kilogram of copper sulfate, 1 kilogram of hydrated lime, 100 liters of water, 2:2:200, 0.5:0.5:50, all the above. So the answer is all the above, because if you see all compositions are same only, just a number changed, but composition is same, right? The next one is the preparation of population affected by the disease is called as disease incidence, disease index, disease intensity, and disease severity. So the answer is disease incidence. Proportion of population by affected by a disease is generally called as disease incidence. Next one, apple or pear blight pathogen having a flagella type. First of all, we need to know what is apple or pear caused by. Irvinia amylovora. So Irvinia amylovora is caused by apple or pear blight, right? So if we see the types of flagellation, Atrachus, Monotrachus, Amphicycus, in the previous slides also we discussed. Irvinia is the best example for Peritrachus flagella. Here it is caused by Irvinia amylovora, which is an example of Peritrachus flagella. So the answer is Peritrachus. The conversion of inflorescence into green leaf-like structures is generally the symptom of phyllodes, Sesamum phyllodes, little leaf of brinjal. It is a phytoplasmal disease generally. So the phyllodes special character is the inflorescence is converted into green leaf-like structure. That is the example that is related to phyllodes disease, which is a phytoplasmal disease. Upper sodium formation is encouraged by the presence of glucose, fructose, dextrose, all the above. So this is generally glucose. If you see rice blast, it produces conidia, right? So this rice blast produces enormous amount of this glucose that helps to penetrate the conidia inside. So due to the presence of enormous amount of glucose that creates an enormous amount of turgor pressure, this pressure create a hole inside the leaf that enters into the plant. So due to the presence of glucose only, it creates enormous amount of pressure. Next one, Epiven is a model used for apple scab. These are all our forecasting models, right? Phylum Plasmodium belonging to the kingdom. This question was repeatedly asked in the previous also. Generally it comes under protozoa. Next one, suspensor can be observed in which of the following sexual spore? Oospore, zygospore, ascospore, and sporangia. So they asked a sexual spore. Forget about the sporangia. It is not a sexual spore. The next one, oospore is a sexual spore that is produced Oomycota group of fungi, where it won't produce any kind of suspensor. Ascospore is also sexual spore, but it is a normal kind of spores, right? When it comes to zygospore, which is produced by Zygomycota group of fungi, it will produce a suspensor. If you see the right side picture, inside a ball-like structure is called, that is zygospore. That was holded by two kinds of holdings. That holded is known as a suspensor. This is the microscopic image of zygospore. So this generally suspensor is produced by zygospore. Next one, pathogen required several years to complete the life cycle is known as. So monocyclic means the pathogen complete one life cycle in a cropping season. So polycyclic, poly means many. The pathogen complete many life cycle in a single cropping season. Polyetic means the pathogen requires 1 to 2 years or more than 2 years to complete the life cycle. So what they have asked is several years, which means polyetic. Okay. Next one, groundnut bud necrosis is generally transmitted by. So groundnut bud necrosis virus is transmitted by. It is a very important one. In the previous year questions also it was repeatedly asked. Thrips, groundnut necrosis virus is transmitted by Thrips tabaci. Then sequence of PCR, polymerase chain reaction, which was developed or invented by Carey Mullis in 1983. For that he got Nobel Prize in 1993. So the process is denaturation, annealing and extension. Denaturation happens at a temperature 94 to 96. Annealing happens at a temperature 55 to 65. But you can take any PCR. The extension will happen at a standard temperature, that is 72 degrees Celsius. This is also important. What is the standard extension temperature in PCR? Such kind of questions also previously asked. So the answer is denaturation, annealing and extension. Answer C, but the standard extension temperature is 72 degrees Celsius. Triple layer membrane is actually present in fungi, bacteria, mycoplasma, virus. See, virus contains no cell wall, right? Instead of cell wall, it contains protein coat. Bacteria, mucopeptide or peptidoglycan cell wall is there. Fungi also contains cell wall. Instead of cell wall, they asked a triple layered membrane, right? So mycoplasma contains no cell wall. Instead of cell wall, it contains a triple layered membrane. So the answer is mycoplasma. Not only mycoplasma but also cytoplasma. So the next one is a flask shaped fruiting body is produced by Pycnidium. Flask shaped asexual fruiting body, you can also consider as asexual fruiting body. Pycnidium, Acervali, Cinema, Clistothysium. Clistothysium is generally a sexual fruiting body. Forget about it. It is a completely closed, right? So Pycnidium, Acervali, Cinema. To understand this, we'll see here. Pycnidium, hollow flask shaped. Sporadium, hemispherical barrel shaped. Acervali, sacer shaped. Cinema, long aggregation of IP along with the answers have been given. Shape of the fruiting body is a flask shaped in Pycnidium. Cushion shaped in Sporadium, sacer shaped in Acervali. Cinema produces look like a branch direct conidiophore. So these are all examples. What they asked is flask shaped, which is produced by Pycnidium. So the answer is Pycnidium. So examples also important. This Pycnidium produces Macrophomina, Paschalina, Fusarium or Tuberculina epicoccum is produces Sporadium. Acervali, Coltrotricum, Cinema means Certosystem or Graphium. So the last one is Neetham reported the first nematode. The world's first nematode is Anguna triticii, that was reported by Neetham 1743, and it is associated with the yellow slime rot, yellow ear rot disease of wheat. That is also associated with a bacterium called Corynebacterium triticii or Clavibacter triticii. This is the first nematode. Is also very important. Anguna triticii is the first plant parasitic nematode reported by Neetham 1743. So these are all the answers related to the questions that we have discussed. So for more information, whatever we are discussing, all those things are the crafts of my book, A Vision into Plant Pathology, a complete student version. For further guidance and doubts clarification, students can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.